Okay, thanks everyone for coming. So I'm going to talk about two measures of biodiversity that are based on trees. There's a particular interesting question that I'm going to look at is what happens to those if there's rapid extinction at the present? So the first of these measures was introduced by Dan Faith. Of course, if you've got a subset of species, you could just count how many species there are, and that's some measure of diversity, but it ignores the fact that those species have evolved on a tree. So Dan Faith's idea was you should really consider what proportion of the tree is spanned by that set of species. So if I take a set S of species at the present, I add up the lengths of the branches between the root and those leaves. So those branch lengths, they could correspond to time, or they could be some measure of how much evolution has occurred along those branches. So we add those links together. So this is called PD, phylogenetic diversity. It's a function from the set of all subsets of my present day species set X into the positive reals. It has some interesting combinatorial properties. If you want to know what is the PD, what is the subset of species that has maximal PD of any given size, that can be solved very quickly using a greedy algorithm. Also, if you want to count how many max PD sets there are of given size, that's also possible using generating functions techniques. That's a recent result of our PhD student, Kerry Manson. What about this problem? Suppose we want to find how bad could it be? You know, what's the maximum amount of PD that could be lost if a certain number of species were to go extinct? That's a little bit of a different problem. We're really trying to maximize that quantity on the bottom there. And here greedy doesn't work because the two species whose extinction would cause the largest loss of PD are those species A and B on the left. On the other hand, for those two species, if just one of them was to go extinct, that would lead to the least loss of PD. So greedy certainly doesn't work, but it can be solved using dynamical programming, so polynomial time. Now there's been a lot of interest in how well PD acts as a proxy for what biologists might perhaps be more interested in, trait diversity or feature diversity. There's been a vigorous debate in the literature, number of papers saying yes, it's approximately similar or no, it's not. I'm not going to go into the details of those here, but I want to just talk about a very simple model. It's a discrete model of feature evolution. So for each species X, we have a corresponding set of set of features. So we think of a species as a bag of features, if you like. So these features are discrete and they are sufficiently complex that we would expect perhaps they only evolved once in the tree. So it could be a gene, a trait, a genomic fragment, or whatever. So how do we define feature diversity for a subset of species today? Well, we simply count how many features are there that are present in at least one of those species in that subset. Okay, so it's the size of the union of those feature sets. You could assign each feature a value, and instead of defining it this way, you could take a sum. Pretty much everything I'm going to say would still be true in that case. Now notice that in this definition of feature diversity, there's no tree at all. And the greedy algorithm doesn't work like it did for PD. If I want to find a subset of size k that maximizes feature diversity, it turns out this is an NP hard problem, so there's probably not even a polynomial time algorithm for that. Okay, so let's think of feature diversity evolving on a tree. So in this example, we have features arising once, but in the case of feature alpha, it also gets lost. So in this case, at the present, those four species, those species at the top there, the feature diversity is 4. All four of those features, alpha, beta, gamma, and delta, are present. Now, if I take a subset of those species, say B, E, and F, the only feature they mention is delta, so the feature diversity of that subset is 1. So this leads to the following question. Given an assignment of features to a set of species, when is there a phylogenetic tree and branch lengths that you can put on that tree so that feature diversity exactly corresponds to phylogenetic diversity? Meaning that for every subset Y of X, the feature diversity of Y is the same as the PD of Y on that tree with those branch lengths. The answer is quite simple. It's true if and only if these sets are nested. So that is the set of species that contain each of the given features. So the nesting is like a hierarchy. An alternative way to think about this is to say it's true if and only if each feature could have evolved on some tree under a novel plus no loss scenario, meaning each feature arises exactly once and is never lost. The crucial words here are could have, because these two scenarios are cases where on the left a feature has arisen more than once, but it could have been described by a feature just arising on the edge below it, a single gain of that alpha. So that is this one here. Similarly, here we have a gain and a loss. That's just the same as a gain. So that's why the words could have are important here. Okay, so what if we allow some loss though? Let's think of this stochastically. So let's think of a model in which features arise at a constant rate along the branches of the tree, but each feature can also be lost at a fixed rate nu. So this number of features then along a path is going to be described by a very classic constant birth linear death process, which was studied, I think, 72 years ago by Feller, and a lot has been discovered about it. The important thing for us is that as time goes on, rather than PD just adding stuff up, this number of features you get approaches an asymptote. And in fact, it's R over nu, the rate of new features divided by the loss converges to a Poisson. Okay, so given a subset of species, we can talk about the phylogenetic diversity of that subset. This will be a random variable, because although the tree here is not random, the process of feature gain and loss along the branches is a random process. I'm going to mostly look at the expected value of that random variable, and we can calculate that very nicely. It's the basis, in fact, of this evo-heritage idea that's described in the latest CIS file. I won't go into the details of that, but there are actually applications to real data in that paper. There's not, unfortunately, in my talk, though. I'm a boring mathematician. Okay, let's think about expected FD. When can it be described by PD? In other words, can we choose branch lengths for my tree so that expected value of FD coincides exactly with PD for all subsets Y of X? Of course, if the loss rate is 0, then what I said before applies, and the answer is yes, for trivial reasons. But in general, that's not the case. If the rate of loss is positive, we have this rather special result. We have this exact correspondence if and only if the tree is of that particular type. So it has to have depth at most 2, and not even all trees of depth at most 2 are in that list of 4 possible types of trees. Okay, so now I want to turn to this question of what happens if we have a sweep of extinction at the present. So the very simplest model is Raupp's field of bullets model, where each species independently survives or not with probability S. So we imagine some rapid extinction event at the present. So in this tree here, the feature diversity amongst all of the species is 5. But when I go to if 3 of them go extinct, now the feature diversity is 3. Alpha, beta, and gamma are the only features that survive. And so I can look at that ratio, 3 over 5, which is 0.6. Now, I'm going to also, we could look at fixed trees, and we've done that. It's kind of more interesting to look at random trees. So classic birth death trees that are used widely in evolutionary biology. You have a rate. You have we have 3 processes going on here. We have a birth death process that generates the tree. On that tree, we have a feature gain and loss process operating, and then at the tips of the tree, we have extinction at the present, with S the probability of survival of any given species. So we can prove this result that this proportion of feature diversity that survives the extinction at the present, it converges in probability as the tree becomes large to a ratio of expectations, and that's really nice because we can compute expectations easily. So what can we say? Well, in the special case where the rate of loss is 0, that's just the PD case. This was analysed some time ago, and you get this behaviour where you have, this is species richness, y equals S. This is for the Yule model, where you have no mu is 0, and as the death rate for the tree increases, it heads up towards the unit step function. So what happens when u is greater than 0? Well, we get this exact result after a bit of work. We can express it explicitly as a ratio of 2 integrals. Unfortunately, we can't solve that integral. And it's low dimensional. And we have this general inequality here that this ratio for FD is always less than it is for PD. So what's the take-home message of this talk for biologists? I guess it's this, that if you're using FD as a proxy for PD, you're probably underestimating. If you're using phylogenetic diversity as a proxy for FD, you're probably underestimating how much diversity has been lost with features. How do I have a minute, or should I sit down? Okay. There's some future directions, and there's some further details if you're interested. Happy to take any questions. Yep.
I know you've done some work on autocatalytic sets. Do they have any relationship with this?
No. I talked about that at another evolution meeting. Okay. Thank you.
Well, thank you so much, everyone, for joining while I talk about some of the work I've been doing in my postdoc with Dr. April Wright. Now, today I'm going to focus a lot on morphological phylogenetics, and to talk about that, I first need to discuss a character. And what a character is it's essentially a feature that exhibits various phenotypes, and it's very, very broad, because oftentimes, if we take a skull like this, you can basically point to anything on that skull, and we can make a character out of it. Because the characters are really made up by their constituent states that express what the phenotypes actually are. And these are often discretized into 0s, 1s, and 2s to ease statistical manipulation when generating phylogenies. And if we want to look at an example of this, we can look at something like the jugal squamosal contact. So this is going to be one of the cheekbones, and if we look in early reptiles, something like this Rugorhynchus, generally we'll expect it to be small and pillar-like and to form the lateral temporal fenestra. Or there may be the derived condition of it being more expanded and eclipsing that lateral temporal fenestra. So in this case, we would see that the jugal squamosal contact is broad based on that red squiggly line, and so it would be the derived state of the condition. Now, this is useful because we can use our transition or Q matrices to generate our hypotheses about how we expect that character to evolve. And in the case of morphology, we're almost always going to be expecting that the forward and backward rates are the same. And this is a very useful system, but I really want to touch on some of its limitations. And the one that I focus the most on is I see discrete characters as being abstractions of reality. They don't actually have any objectivity in them because you can turn anything into a character, really. And if we wanted to look for a little more objectivity, we could try continuous measurements, say taking a ratio or length of a bone or something. And in these cases, it can be easily compromised by ontogeny or fossilization. I probably should have said I'm mostly a paleontologist, so I think about these things really from the approach of what can we take or learn from the fossil record given these works. And continuous variables also suffer from Riemann sum problems where it can be difficult to divide, like where do you actually cut the slice of the pie to be able to identify how these traits are evolving along a tree. And so the kind of go-to method has been to ignore that and stick with the discrete characteristics and assume that there is always an equal rate of exchange forwards and backwards. And this has been the dominant method. This is how parsimony generally is constructed, and parsimony exists as, I would say, the dominant tool in paleontology for about as long as we've been using it. And I want to contrast the way that we think about these morphological characters with the kind of information that we have from molecular characters and from molecular phylogenetics. So unfortunately, we don't have decades and decades and decades of lab and biochemistry studies. We don't have exact rates of transition or transversions like nucleotides do. And DNA is homologous across everything alive. I mean, it's the only thing I know of that you can use to turn a frog into a dinosaur. So it's pretty useful. And in general, these lab and biochemistry studies have allowed for us to be able to modify the transition matrices to follow our expected transitions and transversions between the various nucleotide states. And I view this as there being a thermodynamic limit on rates. We are looking at this at the lowest possible scale, and so we are seeing the constraints that are going to limit how much information we can extract from molecular data. And because of this, molecular data and any study essentially informs one another, as well as all future studies. And you can think of it as this joined homology and simplicity as allowing for nearly infinite number of ways to analyze what is an extremely finite system. And so what I want to focus on is the way that molecular phylogeneticists have been able to account for some of the clear issues that were always going to be a hurdle. And what I really want to focus on today is accounting for rate variation. So knowing that there's differences in transitions and transversions, we expected from the get-go that we would be running into rate problems and how to account for them and how we develop our phylogenies. And because we have so much information, there was never going to be a time it wasn't going to be heavily parameterized. So for about the past 40 years, we've been using the same metric, which is a discretized gamma. And so this is a distribution where we can simply alter the alpha parameter, and it gives a variety of different shapes, so it allows for really good flexibility in assigning rates to your characters. And why this is uniquely effective is it doesn't actually influence the transition matrix. You're able to maintain that in its original state, and instead these are going to be pushing those changes onto the branch lengths, shortening or lengthening them. And when this was first proposed, a gamma rate category of 4 was kind of found to be the best of the likelihoods, and we've just kind of stuck with that. There really has not been critical assessment of why we choose those numbers or if other numbers are better. And so I really want to think about how we can take that molecular information, and I want to envision a Bayesian future for our prehistoric relics and also the fossils that they tend to study. Now, if we consider what the character is and we consider its history, we will always expect variation and homoplasy. But historically, we've attempted to remove as much of this variation as possible from our models and simply present what is simplest. And I think every single person in this room would agree that biology is rarely simple, and that why wouldn't we try to account for that variation that we know is going to exist, and why wouldn't we work to account for it in meaningful ways? And so if you're an aspiring paleophylogeneticist, you'll probably be on a little bit of a pipeline, starting with equal weights parsimony. You work yourself up to likelihood-based modeling, and once you get to Bayesian inference, there's kind of no going back because you have so much flexibility that I don't know that I could go back to that small brain state. So my big question for today, and what I really want to address, is kind of taking a look at paleontological data sets and asking, do these data sets that were created for use with parsimony exhibit among character rate variation, and if so, to what degree? So not even thinking of how to make new characters, but the data that exists, does it have information that assumes variation should be present? So for this, we have a total of 574 morphological data sets. These are almost entirely paleontological. They're compiled by Graham Lloyd on his personal website, and we combed through them. They range in size from 5 to 279 taxa, from 11 to 364 characters, and they broadly sample all of metazoa. So we're not focusing on any one clade. For our analyses, we had a number of parameters that we wanted to keep constant. So we assigned an MK model. You can think of this as simply an equal rates transition matrix. So we're starting our assessment by approximating parsimony. We set an exponential branch length on our prior, or a prior for an exponential branch length. We ran for 100,000 generations in RevBayes. And then we checked our results by IN tracer using an estimated sample size of 200 as our general threshold for whether or not something has reached convergence. Now, to evaluate what was actually, whether or not there's variation, we employed a reversible jump MCMC, so allowing the model to jump between different model parameters entirely to see after the MCMC how long it has spent in each of those categories. And what we were really varying was our alpha parameter. So this is an estimate that will essentially tell us our model preference. So if we find an alpha value of 0, it's going to suggest that there's a lot of variation present in the data, as opposed to an alpha value of 1 times e to the 8th, which is just a very, very big number that essentially says we're not able to find any unique differences between these characteristics in the way that they're transitioning. And we also varied the number of rate categories to try to test that historical question. And to assess some of our effects of this and what's actually going on, we took the trees that we found to have preferred finding variation in the data, and we reran them setting G to 4 and G to 0 to see what happens if we don't include or don't account for variation that we expect to find in the data sets. We compared the result in posterior distributions in R, and we looked at things like Robinson-Fould's distance to see how the actual trees differ from one another. And so to get into some of our results, what we see for our alpha values is we have about 150 data sets that really show strong preference or show that there's a lot of variation within the data. And there's about 250 that show little to no variation. And I think what becomes really important here are these data sets that have very slight amounts of variation but are going to ultimately potentially have a large impact on our trees. When we actually use a threshold model to limit those to whether or not variation is present or not, we find roughly 1/3 of our data have no preference for variation. So 1/3 of our data fit parsimony quite well, and about 2/3 of our data suggest that there is some form of variation, whether it's minimal or a lot. And if we want to look at what that actually looks like, we can investigate the individual site rates. So these are our 4 gamma site rate categories. They're generally, or they're always going to be slow evolving, 2 intermediate states, and then a fast evolving rate category. And there's a large distribution centering around 1 here that is generally suggesting that there may be very, very little variation in a number of these data sets. And so to interrogate that a little further, we looked at the difference between the fastest and slowest evolving rate categories, and we see that for most of these data sets that have variation, it's quite minimal. But I think we should be concerned about these data sets where there are a number of characters that are evolving much more quickly than other characters. If we model rate variation to exist in the data, but it doesn't, we find that all 4 of our rate categories equally distribute around 1, and if you look at the differences between them, they're mathematically indistinguishable for the case of like what we're doing. So the model is able to collapse itself into a single rate model if only a single rate is established. And if we're looking at Robinson-Foold's distance, the way to think about this is a distance of 0.0 is going to mean two trees are identical to one another. A distance of 1.0 would mean they're completely different from one another in terms of their topology. And so we see again, a lot of the time, it's not that big of a deal. You're probably fine to run your analyses. But again, if you simply don't include variation in the rates of change or rates of transitions between your character states, you can get trees that are completely distinct and potentially nonsensical. And I think we should be more concerned about this possibility than the distribution on the left. And in what this actually means is if we fail to account for that variation, we tend to see that most of the time our trees aren't that much different from one another, but in some cases, not accounting for variation will produce a much longer tree. And we find zero support for 4 rate categories. Looks like more is probably better with morphology. So for our data sets, what we've been able to see is that relatively few of our tested data sets have very disparate rate categories. Is that 3 minutes left, or am I? Okay. And when variation is present, most of the data sets are going to exhibit very little variation. But again, and in general, paleontologists have been pretty good at making parsimony. This should be somewhat validating, but again, there's that concern that some of the data sets show much larger differences between the individual rate categories. Now, thinking back to that idea that our DNA transitions are thermodynamically constrained, morphology, on the other hand, would be completely unconstrained. There is no single unifying mechanism that is controlling a bump on the skull versus the length of a femur. And I see this as being, morphology is generally subjective to the researcher and to the available set of taxa, so it's generally going to vary. And so in most cases, we would expect there to be more variation because there are not unifying mechanisms. So in the end, when you don't have variation, the model will find as such. When variation is present, it's often small. And some data sets are more sensitive. But at the end, if you under-parameterize your models, you run the risk of getting nonsensical results that could very much change your inferences. And if you over-parameterize and variation isn't present, the model will collapse in on itself and find that I am done. So thank you very much, and let's get the next one up.
Hi everybody, thank you for being here. Today I'm going to show you a new way to model among character rate variation in morphological datasets for Bayesian phylogenetics. So why morphology? Even though we arguably live in a phylogenomic era, morphology is still almost exclusively the only source of data that we can use to try to place extinct species of fossils in a phylogenetic tree together with extant species, so living diversity. And we know that doing that can give us a much more complete and sort of comprehensive view of macroevolutionary patterns and processes. And as I saw already, like in previous Brennen's talk, usually in phylogenetics, we use morphology as discrete characters. And this is a very complicated and complex process, usually carried out by morphologists that are experts in specific taxa. Brenen talked about a lot of the things that go on when you build such a discrete character matrix. I want to focus on 2 aspects of this type of data that are very different from, for example, molecular data sets. First aspect is that the identity of morphological character is extremely heterogeneous. So here, for example, you can have one character that is like the shape of a bone or like the shape of even like a little part of a single bone, and the other character can be body size, large and small. So the identity of morphological character is extremely heterogeneous. The other aspect I want to focus on is that when morphologists code morphological data into these like data sets, they tend to look preferentially for potential synapomorphism. So they look at variation in a different taxa, and they try to see which are the character states that can be shared derived character states uniting some taxa to the exclusion of others. So in likelihood-based morphological phylogenetics, the building block has been the MK model introduced by Paul Lewis in 2001, or MKV model if you use the correction for variable characters, because most of the times we sample only variable characters for morphology. And this is quite a simple model. You can see here the instantaneous rate matrix, the Q matrix, and mu, the rate here, is the only three parameters. This model has a lot of assumptions going on. I want to focus on only one of the assumptions, and that's that all characters evolve at the same rate. And we might ask ourselves if this assumption is reasonable. Well, if you think about the heterogeneity of the identity of morphological characters, you might think this is a very unreasonable assumption, because if these are so different entities, then they probably also evolve at different speed, at different rates. On the other hand, though, maybe like in the ideal morphological matrix in the mind of a morphologist, that's made mostly of characters that vary only one or few times, and that they define monophyletic clades to the exclusion of others. So if these characters are mostly synapomorphous, then maybe it's a reasonable assumption that all the characters evolve at the same rate. So this is what I wanted to explore in this project using the software RevBase, and there are two main questions that I'm asking here. First, is there among character rate variation in empirical morphological datasets? But second, most importantly, what is the best way to model it? So I'm going to show you, first present like in general how we model a CRV, and a new way to model it that we call to each character their own rate, or Tector models, and we'll see them soon. Then I'm going to show you some results from simulation studies, and then I'm going to show you some results from empirical analysis. So as we saw already in previous talk, usually the way that we go around in modeling among character rate variation in morphological dataset has been co-opted from molecular studies, and that is using a discretized gamma distribution, usually with 4 categories. Here I'm showing you an example with only 2 categories, and you can get an idea of how that works. The distribution can show a lot of among character rate variation or very little among character rate variation, and if you have two rate categories, all the characters are assigned to either one rate or the other. If you have 4 categories, they are assigned to 4 different rates. And we saw in previous talk that maybe a higher number, like 8 categories, might be more appropriate for morphological datasets. And I have to say that people have also used for morphology a discretized log-normal distribution, again, working pretty similarly to the discretized gamma distribution. But the new model the new kind of models that I want to present here, called tectors, or to each character their own rate, the title is pretty self-explanatory. So what I tried to do is to assign basically a Q matrix, an instantaneous rate matrix, to each single character. So each single character gets its own Q matrix, and its own Q matrix can have like the proper number of states for each character, and each character has its own rate multiplier here, mu n, where n is the number of characters. So we actually end up with n free parameters. It's a lot of free parameters, and it's a very parameter-rich model. And there are we tried 2 different ways to sort of model those rate multipliers. And we tried a gamma prior, a continuous gamma prior on the rate multipliers, and a symmetric Dirichlet prior on those rate multipliers. And both of these priors have associated hyperprior for the parameter that governs the shape of this distribution. So we have like alpha for the gamma prior, and concentration, or also called alpha sometimes, for the Dirichlet prior. And you can see that you can have, like, depending on the size of this parameter, you can have like very sort of diffuse distribution or very concentrated. So I'm going to show you now the pipeline that we used for the simulation analysis. We did this to try to understand how these models work under a different set of simulating scenarios, and especially to understand whether the Tector models suffer from overparameterization bias, or whether instead still behave nicely when the underlying generating model is more simple, like a simple MKV model. So we picked empirically derived trees and parameters from previous analysis under 5 different models. These are the simple MKV model, the MKV model with 8 categories gamma, MKV model with 8 categories log normal, MKV with tector gamma, and MKV with tector Dirichlet. And what we do is we simulate 100 datasets per model, and this is the size of those datasets because that's the original size of the dataset that has been used to infer the empirically derived trees and parameters. And then for each of the datasets, we try to estimate topology and parameters using the 5 different models. And then we compare these posterior estimates with the true topology and parameters that generated the datasets under these different model scenarios. So let's go to some results. First of all, here I'm going to show you results when the simulating model, so the true model, is a simple MKV that doesn't have any among character rate variation. So here in this plot, you can see tree length plotted, and at 0 here, at the dashed line, it's the true tree length. On the left, shorter trees. On the right, longer trees. You can see that all the models do pretty much equally well in estimating the true tree length. The tector Dirichlet model slightly overestimates tree length by about 5%. What about tree topology, though? Here I show you some plots of distribution of topological distance calculated by Robinson folds. On the left, you have the true tree. The more you go on the right, you have more distant trees. And here is the percentage of times when the models recover actually the true tree. And these distributions are pretty similar across all models. I would say that the tector Dirichlet model seems to be doing slightly worse. What happens when the true model is a discretized ACRV model like a discretized gamma model? Well, these results for the tree length are very surprising because as you can see, the discretized model actually do a very bad job in estimating the true tree length. This shouldn't happen. And we are trying to understand why that's happening. There might be an issue with the implementation of the variable character correction for the discretized model specifically, but we're not able to figure out exactly what's going on. In any case, the tector models do really well at estimating tree length, even when the generating model is a discretized ACRV model. And if you look again at topological distance, these distributions are pretty similar, but the tector models do slightly better in estimating the true tree. When the true model is the tector models do really well in estimating the tree length, and all the other models are subpar. And again, if we look at the distribution of the topological distance, these distributions are pretty similar, but the tector models and the discretized log normal models seem to be doing slightly better. Okay, so now we saw the behavior of these different models under different simulating scenarios, and we have seen that the tector model seems to be performing pretty well under a wide variety of scenarios. We want to see, though, if these different models make a difference. Do they really matter when we go analyzing empirical datasets? So what we did, we picked 88 datasets from the literature, varying from around 5 to more than 200 extant taxa, from 15 to more than 1,000 variable characters. We analyzed them under the same 5 different models that I showed you until now. And we get some tree estimates and parameter estimates, and we can compare them, and we can see if applying different models really makes a difference for real empirical datasets. So I'm going to show you now some plots about among character rate variation, about the estimated parameters for these different empirical datasets that govern that shape, that tells us if there is some among character rate variation or no among character rate variation. So here, for example, is the discretized gamma model. On the left here, you have datasets with among character rate variation, and on the right, no among character rate variation. And we can see that slightly less than half of the datasets analyzed have some evidence of among character rate variation. Interestingly, with the discretized log normal model, that percentage is quite higher. But again, you have a similar distribution of this parameter, like very distinct, like the ACRV and no ACRV datasets. Tector gamma model does pretty similarly, but not identical to the discretized gamma model. And here is the concentration parameter for the tector Dirichlet model. This is quite different because you don't see this clear distinction. Instead, it's more of a continuum. And we can actually see what that looks like in detail. On one end of the spectrum, we have datasets such as this one. Here I'm showing you per character relative rates, and histograms, these are like counts of the different characters. So you see in this dataset, all the relative rates are close to 1, so it means no among character rate variation. In datasets like this one, for example, we can have like 3 to 4-fold among character rate variation between the slow evolving characters and the fast evolving characters. And then we have datasets like this one, where we have 1 order of magnitude or even more of variation between the slowest evolving characters and the fastest evolving characters. Okay, what about tree length? Of course, here I cannot compare tree length with a true tree length because we don't know the true tree. But I can compare it with models. So here are tree lengths compared to the MKV model, and we can see that for many data sets, all the models see a similar tree length or same tree length, but for some data sets, it does make a difference, and there is different tree lengths. So on one of the end of the spectrum, we have data sets such as this one, in which all the models show the same tree length estimate, and on the other end of the spectrum, we have data sets such as this one, in which models clearly estimate very different tree lengths, and we have reason to believe, thanks to the simulation studies, the detector models are doing a better job in estimating the proper tree length. Okay, I showed you tree length. What about topology? So here I'm showing you again topological distance from the MKV model, and again, a good bunch of data sets don't show any difference, but some do. And this is difference not only between MKV model and all the models with among character rate variation, but also between the among character rate variation models. So for example, discretized gamma and the other models, we see difference. So we do see that for some data sets, applying the detector models really impacts also the topology. So as a summary, we are seeing that among character rate variation is present in about half of the discrete morphological data sets that we analyze. We developed and implemented in RevBase a new class of models to model among character rate variation for discrete morphological phylogenetics, and these are tector models to each character their own rate. And tector models can properly model ACRV that is observed in empirical data sets. Most importantly, they do not suffer from overparameterization bias because they can collapse quite nicely to the simple MKV when there is no among character rate variation in the underlying data set. And I think I've showed to you that they do a pretty great job in estimating tree length under a different set of scenarios. And I think these are quite exciting models to develop from now on to try to include more complex and more realistic models of morphological evolution, such as, for example, unequal state frequencies. I'd like to thank funding bodies, the LMU, and the HENA Lab, and thank you for your attention.
I'm going to present some work that I did this year with my advisor, Dr. Vika, and it's really a kind of an extremal problem. Just with the shape of trees. And so I'm going to motivate it a little bit. So to kind of zoom out, stairs 2 is a balance index, and I'll tell you more about what that is. But the reason we want to study that is we've got models for how trees should evolve in time, and that'll give you a distribution of trees with probabilities. But we've also got a bunch of trees that have been inferred by people who are giving talks at this conference about the best way to infer trees, and so we want to compare those distributions, but there's tons of trees, so that's hard. And so what we want to do, one idea is, we'll throw them through a function that assigns trees a number, and we'll try to make the expectations look the same, and then hopefully you've improved the model for how the trees evolve. And so one thing that people notice when they look at trees generated under these models and trees that have been inferred is that, okay, the ones under the model look like they're probably on average kind of more balanced. So we're interested in a way of quantifying balance as well. And then just another little bit of motivation, particularly for stairs 2, and this is a little backwards because this was discovered after, we found out about it after we did most of the work, is that stairs 2 is particularly good at distinguishing between trees from constant rate birth death models, so the Ewell model, if you're familiar with that, that's the basic one for that, and empirical trees. So what that really means is that we should think that stairs 2 is probably telling us something particularly good about how the models are wrong. So okay, this is the structure we're dealing with. I mean, if you were here for Dr. Steele's talk, he was talking about rooted binary trees as well. It's critical that it has to be rooted and binary for stairs 2. So yeah, there's the root. It's all going down. So unfortunately, I did it the opposite direction on Dr. Steele's talk. And then I'm going to call the tips leaves. That's what I've become accustomed to. And n is always going to refer to a number of leaves and what I've got going forward. Okay, so to quantify balance, we want to set not we, but it's been set before. You want a baseline for what you have to be a balance index, but it's in general ambiguous. So this baseline is that you've got to say for each leaf number that the caterpillar tree, which looks like this, something splitting always on the same side each time, that's going to be the least balanced. And then whenever you've got this structure like this, a fully balanced tree, you got to say that's the most balanced. And that makes, I think that's clear why that makes sense. And so this leaves, the natural thing this always leaves open is that, well, the caterpillar exists for any number of leaves that you want, but the fully balanced tree only exists for powers of 2 because it's got to be perfectly balanced. So that's in general not known for balance indices, what the best tree is when you don't have a power of 2 for the number of leaves. All right, so now I'm going to keep dancing around, and now I'm going to tell you what stairs 2 does. So stairs 2 is an index like many other balance indices, but not all, where you've got a sense of balance at a particular interior vertex, and you're going to aggregate it in some way. So if you're familiar with the Colas index, that kind of, the sense of balance at a particular vertex is you take the absolute difference of numbers of leaves on each side below it. So stairs 2 is different. We're going to, instead of taking the difference, we're going to take the ratio, which ratio, so that min over max is saying, okay, I'm going to get the ratio that's less than 1. And then we're also going to take the average at the end. And so because for this work, we're only comparing things with the same number of leaves because I'm only interested in the best tree for each leaf number. That part isn't going to be so important. So in some stuff I show later, you're going to see n-1 and stuff of that form kind of multiplying everything out, because what I'm really interested in general is the sums of all those ratios. And so I'm just going to show you a little bit of how this works to emphasize one real thing here. Okay, so here are the interior vertices, everything that's not a leaf or everything with stuff below it. And then here are the ratios for each of them. So on the right, you can see, well, it's all symmetric over there, so you're going to get a bunch of 1s as your ratios. Also at the top, because it doesn't care about the shape underneath, just the number of leaves. And then on the left side, you can see some unbalanced stuff. And if you really wanted to add it up, there it is. But that's, of course, not really the point. We're interested in what we can do for all trees, for all leaf numbers. Okay, like I said, the optimal tree had not been known for trees that are leaf numbers that are not power of 2. But we figured it out, and I called it the binary echelon tree, and I'll tell you what it is. One cool thing real quick is that it is unique for each leaf number. This is not generally the case with every balance index. Okay, so what does the binary echelon tree look like? I think the first way to think about it is you take your leaf number, you're going to take the biggest power of 2 you can squeeze inside that number, put it a fully balanced tree off to the side over there, and then keep going down the line. Right, for 11, you can see the 8, and then you got 3 left over, so you do 2 and 1. And so if this seems familiar, this, of course, is how you get the binary expansion of a number. So you can think of just doing the binary expansion, but what's important is you got to put it, got to put the trees kind of in the right order. You got to join the smallest up to the biggest. And so the first thing that I said is really kind of the key. And so just to emphasize that, what you want to think of a binary echelon tree is it's a big, fully balanced tree, relatively, and then a relatively smaller binary echelon tree. And that'll tell you all of the binary echelon trees, just because there's only one option when you get to small enough leaf numbers. So you can build the whole thing up from that statement. Okay, so you may have noticed on the previous slide I said it's a nice recursive structure. So cool, I've got some recursive identities. They look terrible. But I promise you they're actually nice. It's really just, it's the multiplying out stuff that makes it take up more space. So this first one is the more important one. And so what's going on with this is we're dividing by dividing out by the total number of leaves at the end. So don't worry about the thing at the beginning. So, okay, let's see if I can aim this well enough. Okay, cool, I can. So if I've got a tree of the form, a number, and I'm going to express it as a big power of 2 and then, some leftover. The binary echelon tree looks like this because the power of 2 is bigger, so it's going to go off to the side on its own. There's 1 less than the number of leaves for the number of interior vertices. They're all perfectly balanced, so you're going to pick up a 1 for each of those, so you got a 2 to the k minus 1. And then over here, this is just stairs 2 of the binary echelon tree because that's what the structure has to be for this to be a binary echelon tree. It's got to be a fully balanced and a smaller binary echelon. This is multiplying it out to get rid of the average. So that's just the sums. And then we got the root, and, well, we know that n is less than or equal to 2 to the k. So then we get that, divide by the right thing. There you go. And then this other one, this is if you want to double the number of leaves. You also get something nice because to double the number of leaves to get the corresponding binary echelon tree from the previous one, you just take all the leaves and you split them into 2 at the bottom. The nice thing about that is that it doesn't change the ratios because you just doubled the numerator and denominator for everything up here. So we have the same sums for everything in the original purple tree up here. And then now we've got all these leaves become interior vertices. How many of them were there? There were n, so we got to add n ones. So there's the original stuff, and then the n ones, divide by the right thing, and there you go. So if you want to know the maximum value, you can essentially do that first identity over and over again because that's all a binary expansion is for a number. You're adding progressively larger and larger powers of 2, specifically larger than what you had before. I guess that's important as well. And so I'm going to argue that you can kind of see both of these identities in this graph I've made. So on the horizontal axis is just the leaf number, and then the maximum stairs 2 value for over all of the trees for that leaf number. I've left out 1 because it's arguably not a binary tree, but if you read the paper, you will see that we've given it a conventional value of 0. It makes stuff work nice with the multiplying stuff out with the way we did the proof. But okay, all the powers of 2 are up at 1, right, because everything's balanced in them. And then these red ones are the ones that are halfway between powers of 2, so 6 and 12 are the only ones, and 3, yeah, of course, on here. And so this pattern of them trending upward, this is coming from the doubling one, where it's getting diluted by all the ones at the bottom, so that's why it's going farther and farther up. And then the other identity, I think, is best seen from this, going from like this 1 down at 0, 1, 2, 3, 4. So it goes sort of zigzag pattern. And we add to 4, right? We've got this zigzag pattern again. So we 0 up to 2, down to 3, up to 4, and then down to 5, up, and the same sort of pattern is getting squished up towards the top for the same reason, kind of dilution stuff. All right. And then lastly, I just want to acknowledge, this is mainly acknowledging my advisor because I haven't met the other people, but they've written a really nice R package that made it so I can write 10 lines of code to brute force this for small n and see what's going on and then formulate conjectures. So I just wanted to give it a nice shout out. And then I apologize for the way that I did the on slide citations. I tried to change it, but I messed up all the figures because it's using all the space that was there. So these were the papers that I cited. But that's all I had scheduled for you. So if you have any questions, I'd love to answer.
Have you looked at the stairs 2 index for random trees like rule type trees or PDA?
No. So I haven't started looking at the like the expected value under those models or anything. I have a slight suspicion that maybe the recursive structure might help, but then you'd have to adjust for how 16 versus 1 is going to occur less than 15 versus 2 or something more reasonable. But, yeah. All right. Yeah, I did have a couple other things prepared. So just I wanted to show you the one particular thing at the end. Again, if you're familiar with the coalesce index, this is just the first example where they're going to disagree. Just to emphasize that they do disagree, it does not keep the trees in the same order. So these are the three root topologies for 5 taxa. And then over at the left caterpillar, you can see it's the worst for each of them. Coalesce, by the way, bigger value is worse balance. And then the difference is the coalesce index says the one in the middle is the best, which it seems reasonable, 3 against 2. But then stairs 2 says the one on the right is the best because it's kind of can get penalized for vertices that are not symmetry vertices a lot more. Yeah. That's it.
Start. I don't mind having extra time. Hi everyone, I'm Michelle. I am a PhD student in the Parents Vicucci Lab at the University of Toronto. And I just want to say first that I'm a little intimidated to be here, but I've really enjoyed learning from all your talks, so I hope that you'll get something out of my talk or find it interesting, even if it's not hardcore phylogenetic theory. Okay, so a fossil is a snapshot of the past. And when the fossil record is relatively complete, it's like a film reel, and we can rewatch the tape of life with exquisite clarity. One such exceptional fossil record tells the story of Dendraster sand dollars. Dendraster is the only genus in the sand dollar family Dendrasteridae, found living today on the Pacific coast of North America from Alaska down to Mexico. And in case you did not know this, sand dollars are a weird, highly flattened echinoid. These guys are unique in that most sand dollars are deposit feeders that lie flat on the substrate, but Dendrasterids can stand upright and feed from the water column as well. And this likely has a lot to do with their eccentric morphology that is essentially unique among all sand dollars, where the apical system is out of the center or eccentric to the posterior. They also have an extensive fossil record, as sand dollars have been blessed by evolution to be extremely fossilizable. And the Dendraster fossils in this region are so abundant and well preserved that you can trace their evolution from the group's origin in the late Miocene of California through their spread along the coast to where they're still living even today. And what's really great about them is that you can get fossilized populations. And so in theory, you could evaluate variation within and between species across geographic ranges and time spans, or long time spans, and maybe even over ontogeny. So all of this coupled with insights that we can get from the living representatives on their ecology, behavior, and development make them a great system for testing ideas at the interface of micro and macro evolution. So for my PhD, I'm hoping to get at some of these ideas, but to start, we should probably know about their relationships to inform our expectations and act as a backbone for future analyses. And unfortunately, phylogeny for them didn't really exist yet, and as it stands, the taxonomy of Dendraster is kind of stale. The last treatment was by Rich Moy in 1997, and only looked at the 3 living species, and called for the inclusion of the fossil taxa, which is like maybe 10 or more so species, in future analyses to better understand the history of this group. One of the early attempts at drawing out these relationships was by Q in 1920, and the rest of our understanding comes from various species descriptions in the 19th and 20th centuries, and various statements made by various researchers over the years. But it seems that we have reached a few general conclusions, which I will go over just briefly right here. One is that the genus Merriamaster, which has a few miniaturized pedomorphic species, likely originated from within Dendraster rather than being sister to it and should be synonymized with it. Another is that this genus Scutalaster has been moved to a different family, Echinarachnidae, and we will talk more about them later. These relationships between Dendraster eccentricus, diegoensis, and venturaensis are still solid, or at least they haven't been overturned yet. And these 2 pairs of species have been synonymized. Many other relationships have not really been addressed since they were put out there, and there are species that have since been described or are just not represented in here. Where do they fit into all of this? And finally, most of these statements pertain to shallower nodes in the tree or more tipward relations, and a lot less has been said about the deeper relationships in this group. So is it really such a linear sequence, or is there more branching, and where would the branches go if so? I'll also say that there aren't a lot of people working on this right now, and a lot of these changes are officially agreed upon. So I'm going to be using some of the old names, even if they've been synonymized, for brevity in the rest of this talk. The last thing I'll say on this is that the placement of Dendraster itself within the sand dollar family tree has historically been a point of contention as well. This is because their eccentric morphology shows up quite abruptly in the fossil record, and there are no transitional forms leading up to it. And it's highly likely that this abrupt change was real, since this morphology could arise relatively easily due to a pretty simple modification of the developmental process. But regardless, it has still made it difficult to place them historically. But a general conclusion from morphology is that Dendrasteridae should be close to Echinarachnidae, which is represented by Echinarachnias parma here. And this is corroborated by molecular phylogenies in recent years, including by some people who are here today, that show Dendrasteridae being near Echinarachnidae and a few other families. But I'm going to be focusing on Echinarachnidae for a moment, as this family has historically been used to approximate more or less what the ancestor of Dendrasteridae could have looked like. And also because it has two fossil species that have independently evolved some slight posterior eccentricity, suggesting that this group has the capacity to generate this morphology even recurrently. And so including these taxa could give us some further insight into trait change within Dendraster. So just to summarize the main goals that I'll be addressing today, first is, can we get some clarity on the relationships within Dendraster? And since most of these representatives are fossils, we're going to be making a morphological phylogeny. And because, well, there are a lot of challenges involved in making a morphological phylogeny, or any phylogeny in general. But rather than waving it off or sinking into despair, maybe we can look closer at the data and the characters themselves to see where they might disagree or where any weirdness comes from, because therein we may find the very interesting evolution and maybe come up with ways to work around it. And so to start, I built this phylogeny using morphological data that I obtained from the literature. And as a quick aside, I started this project last year in the first year of my PhD when I didn't even have 2 sand dollars to rub together. So to start on this without any specimens, I turned to the lit. And I later corroborated my coding with specimens that I worked with in a very recent museum trip. And we got thousands of images on this trip, some of which you will see later today. But I'm going to be beefing up this analysis with data that I collect myself from these specimens. As a first pass, the literature data should be okay. And I used 33 characters, taken from a variety of species descriptions and monographs, et cetera. And I incorporated 28 species, so that's most of the Dendraster species, but I excluded some of the less thoroughly described ones, and I still kind of think that that's too many species and they're not all real. And I incorporated most of the Echinarachnid genera with 2 representatives or so each for comparison purposes. And finally, unfortunately, I'm still in my small brain phase, so I used parsimony and paup, but I just wanted a quick parsimony tree to see what the majority signal was. And in the future, I will be implementing likelihood and or Bayesian-based methods for a future tree. And also, you'll be seeing the consensus trees that I got from this heuristic search. So what does it look like? Well, here it is. And first, you'll note that, and I think that the scientific term is that it is polytomicity, especially for these deep relationships within Dendraster and within Echinarachnidae, which is on the lower side. I'm going to draw your attention to a few other particular things. These guys at the top are the Merriamasters, those miniaturized repetomorphic species, and they're clustering together, which would make sense. Here also is a cluster of Dendraster venturensis, diegoensis, and eccentricus. This corroborates the idea that they are closely related, and maybe even that one or both of these above species are ancestral to eccentricus. And so, seeing this tree, I want to understand why it looks like that, both to address it and make a better tree in the future, but also to infer, or attempt to infer, what evolutionary processes might be underlying this, because that's very interesting. And one potential reason would be conflicting character histories. We can start to get at this by looking at character compatibilities. In this initial exploratory analysis, we identify sets of characters that are internally consistent with one another and form major factions of support. Sometimes you'll have traits that disagree with each other. For instance, traits 1 and 2 cannot be mapped to the same tree without homoplasy. In both of these topologies, there's an instance of homoplasy. So that makes them incompatible. But other traits can be mapped to the same tree without invoking any homoplasy. So in this case, traits 1, 3, and 4 are all agreeing with each other while disagreeing with trait 2. We can go through all of the characters and make pairwise comparisons between each of them to see if they're compatible or not. You could conceptualize it something like this graph, where each dot is a trait, And if you map the patterns of compatibilities between characters, you might start to see sets of almost discrete clusters. And these clusters can be thought of as the major factions of support for competing or conflicting relationships in the data. So what do we see? What clusters do we get? Well, here it is. You don't have to read all of this. It's a lot. I will tell you what you need to know. The bolded character names are characters that are invariant within Dendraster, so they don't vary, and are mostly used to distinguish among the families and the genera. And you'll see that they're mostly hanging out in clusters 4 and 7. So there's a bit of disagreement between them, but it seems like less disagreement than if they were evenly distributed among all of the conflicting clusters. All these other characters that are not bolded are variable within Dendraster and typically within the other genera as well, and they're not good for delineating these larger groups. So ideally or typically, they would be helping us to resolve relationships within Dendraster, but they're not doing that job because they're disagreeing. I'll be focusing in on these few clusters as exemplars. Here's a tree made from only characters that are contained in cluster 4, and you can see that they mostly separate Dendrasteridae from Echinarachnidae, mostly. And that makes sense. They are invariable within Dendraster. But compare that to this tree made from only characters that are found in cluster 2, and recall that these are more variable within Dendraster. And these are a lot of characters pertaining to overall shape and some surface or petaloid characters. I'll draw your attention to the fact that Echinarachnius aliscensis and Scutalaster oregonensis are hanging out near some Dendrasters in this tree. And remember that those are the two independently evolved eccentric species. This makes sense. They tend to resemble Dendraster in some aspects of their general shape, but they're still separated from them by other characters that are informative at a higher level. As you can see, they're outside in this previous tree. I will also point you to these two groupings here, because if you look at this other tree made with a different cluster of these lower level variable characters, they have been moved around. So it seems that the competing or disagreement between these clusters of characters may be underlying these polytomies and our lack of resolution, especially for characters that are variable within Dendraster. So what's up with these characters? Why are they so variable? Why are they in disagreement? One potential explanation could be evolutionary lability or differences in it. For instance, overall shape is not only variable between species, but it's also highly variable within species. And actually that has been plaguing me. So if you want to talk about variation within species and in phylogenies, I need to incorporate that. But something like the number of plates is constant enough to be a distinguishing character among the genera. And in this particular case, it's likely because sand dollars with a flat edge like these ones develop their full complement of adult plates at a relatively early stage in ontogeny, and later growth involves accretion around these plates, creating growth rings. And so this likely allows for more variation in overall shape than plate number, generating these differences in lability. The other characters that tend to be highly variable within Dendraster are generally traits that vary over the course of ontogeny, which could also make them more labile and prone to modification by things like heterochrony, for example. Another potential explanation for this pattern would be characters that were polymorphic in the ancestor being differentially sorted among the descendants, something similar to incomplete lineage sorting. In fact, many of these highly variable characters are not only variable in one species, but are found recurrently through a few different species. In this example, Dendraster venturensis and eccentricus are believed to be closely related to one another, and you can see that they share some of these major categories of different overall outlines. So to conclude, I'm hoping to integrate these together and use them to inform my future analyses when I look into more regarding these characters, and ultimately when I attempt to make a better phylogeny using my own data, incorporating stratigraphy and geography, incorporating likelihood and Bayesian-based methods, and I didn't mention it right here, but I would love to incorporate the intraspecific variation because it seems very prominent in this group, and also, rate variation as we've discussed today. So I'd like to thank you all for being here and listening, and I would like to really thank all the members of the Parents Fukuchi Lab for helping me workshop this talk. I had 1 week between coming home from the trip and going to evolution, and I'd like to thank my funders. I'll take any questions if we have time, and if you have any questions, thank you.